Hi, I am Merel Stemerdink, a PhD candidate at Radboud University in Nijmegen. In my PhD project, I focus on the development of a therapy for Usher2C associated retinitis pigmentosa. In this video, I will tell you more about specific research that we conduct. I'm aware that some of you may already know a lot about the Usher syndrome, whilst for others it may still be very new. And for that reason, I would first like to start with a very basic introduction about this topic, before moving over to the details of our research. Different types of the Usher syndrome exist, but in general it is characterized as hearing impairment, a loss of vision due to retinitis pigmentosa, I will tell more about this later, and with or without the balance problems due to vestibular dysfunction. But this is not the case for the Usher syndrome type 2C. So, the Usher syndrome is a genetic disorder, and that means that the cause of it lies within our DNA. For type 2C specifically, this is because of errors, which we call mutations, in the ADGRV1 gene. How does this work? I always like to explain our DNA like it is a recipe book, with different chapters, which are the genes, that explain how to produce different proteins for the function of our body. Let's say that each gene is indeed a chapter explaining how to produce a protein, and a normal functioning gene just perfectly describes how to make a functional normal protein. But once mutations arise, there are errors in this explanation, and therefore non-functional proteins are being produced, or no protein at all. Proteins are the building blocks of our cells, and for the Usher proteins specifically, which are made by the Usher genes, they are mostly important for the cells in our inner ear and for the cells in our eyes, the photoreceptor cells. On the left side of the screen you see an inner ear hair cell, so a cell which is present in your inner ear. And next to it we have the two types of photoreceptor cells, the rods and the cones, the light-sensitive cells of the retina of our eye. And in the blue dots they represent the locations where the Usher proteins are important for the functioning of these cell types. Normal functioning inner ear hair cells have stereocilia at the tips of their cells. These stereocilia are motile, and that means that when there is a sound stimulus, these stereocilia are able to move, and upon movement the cell signals to your brain in order for you to register this sound. And the Usher proteins are especially important for the development of the stereocilia. And in case of Usher hair cells, where these proteins are absent, the stereocilia are not developed well anymore. This is causing problems in the signal transduction after a sound stimulus, and therefore it is causing the hearing problems. Similarly, the Usher proteins are important for the functioning of the photoreceptor cells in your eye. So once light enters your eye, it falls on the back of your eye where the retina is located. Over here I show to you a histological figure of the retina. And as you may see, it is built up by different layers of cells. And one of these cell types are the photoreceptor cells, which are light sensitive. So the Usher proteins are also here important for the functioning and for the maintenance of these cells. And once these are absent, this results in the gradual degeneration of the photoreceptor cells. And this condition is called retinitis pigmentosa. So it is the progressive degeneration of the photoreceptor cells in the retina of your eye. As a result, you may start to experience night blindness, and as it progresses, eventually you will develop a tunnel vision until one becomes legally blind. Different types of the Usher syndrome exist, and they all differ in the severity and the age of onset of the symptoms and the absence or the presence of the vestibular function, which is sometimes causing the balance problems. Also, different genes are involved in the different types of Usher syndrome. The most prevalent type of the Usher syndrome is the type 2, and that is also the main focus in our research group. Erwin van Wijk is our research group leader, and he has already been involved in this research from 2004 onwards. In the past decades, in his research group, we have been able to develop even a preclinical therapy for Usher2A associated retinitis pigmentosa, and this drug is currently in clinical trial. And based on these successes with the Usher2A research, he thought, can we also expand on this for the other Usher2 genes? And therefore, in 2020, I was able to start my PhD project, in which I focus on the development of a therapy for Usher2C associated retinitis pigmentosa. When we look at the disease progression for Usher2C specifically, one is often born congenitally hearing impaired, and that may be compensated by using hearing aids or cochlear implants. Around puberty, one will start to experience the first signs of retinitis pigmentosa, and for this currently no treatment exists. As this progresses, eventually by the 5th or 6th decade of life, one will become legally blind. But in the meantime, we have this window of opportunity. Because the retinitis pigmentosa is of a progressive nature, our goal is to develop a therapy to halt the further progression of ADOGV1 associated retinitis pigmentosa. So the earlier we can therapeutically intervene, the better, and therefore we aim to prevent that people with Usher2C will become legally blind. So what do we need to develop a therapy? First of all, we need to have a strategy, and secondly, we need a model in which we can test the therapy that we have generated. So in my research, I focus on 2 types of strategies. And the first one tries to deliver a healthy copy of the ADOGV1 gene. Secondly, we focus on a strategy in which we try to remove the specific parts of the ADOGV1 gene that have been mutated. In the next slides, I will tell you more about these 2 strategies. The first strategy focuses on the delivery of a healthy copy of the ADOGV1 gene, and we call this gene augmentation therapy. So remember that the Usher syndrome is caused by non-functional ADOGV1 protein or no protein being produced at all, and therefore we aim to deliver a healthy copy of the gene so that the protein production is being restored. And for the delivery of this gene, we use a viral vector, which is like a vehicle that is able to deliver the ADOGV1 gene to the photoreceptor cells in the eye. But there is one big problem. The ADOGV1 gene is one of the largest genes in the human genome, and therefore the gene is too large to fit into these viral vectors for the delivery. So we tried to come up with a solution, and we thought, can we miniaturize the ADOGV1 gene in order for it to fit into the viral vector? So we did this by selecting the specific parts of the gene of which we think it contains the most important information for building a smaller but still functional ADOGV1 protein. Let's have a look at this in more detail. Over here I show a schematic representation of the ADOGV1 protein. And proteins are built up by different protein domains. And once we have a look at ADOGV1, we see that it is consisting a lot of these orange-red dots, meaning that it is consisting of a lot of repetitive protein domains. Our hypothesis is that we do not need all these orange-red dots for it to function properly. And based on the principle that I showed on the previous slide, we have generated different mini-genes that would encode different smaller ADOGV1 proteins. For this we made use of different bioinformatic tools and we made use of conservation scores, but in the end we have been able to generate 3 different mini-genes. The benefit of our first strategy, so the mini-gene augmentation therapy, is that once it appears to be effective, we can treat any type of Usher2C patient with it, irregardless of where the mutations are located because we just simply deliver a healthy copy of the ADGV1 gene. The second approach, to skip or remove mutations from the ADGV1 gene, differs because this is a mutation-dependent approach. Previously, we have already shown that this is effective because the Usher2A drug, which is currently in clinical trial, ultraversion, is based on this second principle. I will explain more about this in the coming slides. Let's go back to the background. I already explained that we can see our DNA as a recipe book, with the genes being the chapters explaining how to produce a protein. But once we zoom in into a specific gene, we can see that it is consisting of different exons. And you can see these exons as like they are the words in the recipe, so they contain the important instructions on how to produce a protein. In our second therapeutic strategy, we specifically target mutated exons in the ADGV1 gene. So let's say that in a healthy individual, exons X, Y, and Z carefully explain how to produce a functional protein. But once there arises a mutation in exon Y, your body cannot read the rest of the recipe anymore, and therefore no functional protein is being produced. We aim to restore the protein production by just removing the single exon, so just generating a recipe missing one word, but then your body can read the rest of the recipe again on how to produce a protein. We hypothesized that by skipping one exon, your body is able to produce a slightly shorter but still functional protein. Now that we have developed these two types of therapies, so the mini-gene augmentation therapy and the exon skipping therapy, we next want to evaluate, are these indeed therapeutic? So are we able to restore the function of the photoreceptor cells in our eyes? And for testing this, we need a functional model. Remember that we specifically want to treat the retinitis pigmentosa, so the eye condition. And the benefit of treating an eye is that it is an immune-privileged organ, and also it's easier accessible than, for example, the inner ears. More importantly, the fact that retinitis pigmentosa is progressive means that by an early treatment we can halt further progression. But then we need an eye model to test our therapeutic strategies. So cellular models exist, or organoids, which are like organs in a petri dish. But unfortunately, we still need animal models to test the visual function. So to test the effect of our therapies in a model that most closely resembles the human eye. In research, often mouse models have been used. And as a result, researchers also previously tried to generate an ADGV1 mouse model. And these mouse models with mutations in the ADGV1 gene resulted in deafness, but strangely they did not show the retinal phenotype. So for that reason we cannot use the existing mouse models to test our therapies. So we need another model, and in our lab we use zebrafish. All Usher genes are conserved between human and zebrafish, and this means that the zebrafish also has an ADGV1 gene in his genome. And when we look and compare the protein structures, we also see that these are highly similar in human and zebrafish. For that reason, we hypothesize that by creating small mutations in the zebrafish ADGV1 gene, we are able to produce a zebrafish model that also shows retinal dysfunction. How to create these mutations in the ADGV1 gene? Well, for this, we make use of the CRISPR-Cas9 methodology. And we can see CRISPR-Cas9 like it is a molecular pair of scissors. CRISPR is able to find the right spot in the DNA where to make a specific cut, and the Cas9 is then able to actually make the cut in the DNA. So once a cut has been made, your cell will try to restore this, to fix this. But often cells make mistakes, and in this case, often mutations arise in these specific genes. And using CRISPR-Cas9 methodology, we have been able to generate mutations in the zebrafish ADGV1 gene. And then it is the question, does this indeed also result in non-functional proteins being produced? In the next couple of videos, I will show you how we use the zebrafish model in our zebrafish laboratory. This is the zebrafish facility in Nijmegen. This facility offers housing, care, and room for experimentation for us as researchers that make use of the zebrafish model. Over here we show a tank with adult zebrafish. But to make a new mutant zebrafish model, we start with zebrafish eggs. This is because we inject the CRISPR-Cas9 mixture to generate mutations in the ADGV1 gene already at the single cell stage. After injection, within 5 days, the eggs grow into zebrafish larvae. In total, it takes 3 months for the larvae to grow into adult zebrafish. Following this procedure, we have been able to generate our mutant ADGV1 zebrafish model. So now that we have generated mutations in the zebrafish genome, next the question arises, what happens to the protein production? So as a recap, remember that the ADGV1 protein is especially important for the function of the photoreceptor cells in the retinas of the eyes. And similar to the human situation, zebrafish also have these photoreceptor cells in their eyes. So we made retinal sections of the zebrafish retinas. And we compared sections of healthy zebrafish with retinal sections of our mutant zebrafish model. So in the upper section we see a retinal section of a healthy zebrafish. And we made use of a specific technique which is called immunohistochemical stainings. And therefore we are able to specifically stain for the presence of, in this case, the ADGV1 protein. And as you may appreciate, there are little red dots visible in the healthy zebrafish retina, indicating that indeed the protein is present. But when we compare it to the lower picture, to the section of a mutant zebrafish model, there are no red dots present anymore. And this indicates that ADGV1 is absent in the retinas of the ADGV1 zebrafish model. So now that we know that the ADGV1 protein is absent in the retinas of our mutant zebrafish, the question arises, what is the consequence for their retinal function? Using ERG analysis and using the setup shown over here, we are able to track the activity of their retinas in response to a light stimulus. So more specifically, we can track to what extent the light-sensitive cells, the photoreceptor cells in the retina, are able to convert a light stimulus towards an electrical signal. So we track the electrical signals sent by the photoreceptor cells. And in green we have shown the electrical signal sent by a healthy zebrafish. And in red we show the electrical signal that is being produced by photoreceptor cells in our mutant zebrafish. And what is important to know is that the height of this B wave amplitude is indicative for the retinal function. So the higher this amplitude, the better the photoreceptor cells are able to convert a light stimulus towards an electrical signal. And as you may appreciate, the lower B wave amplitude for mutants means that there is an impaired retinal function in our mutant zebrafish model. So as a wrap-up, in this video I've introduced you to the Usher2C research that we conduct at the Radboud UMC in Nijmegen. In the past years we have generated ADGV1 mutant zebrafish. And this is the very first animal model that allows us to study ADGV1 associated retinal dysfunction. Next it is the question, using our therapeutic strategies, are we able to restore the protein production and are we able to restore the visual function of these zebrafish? I have introduced you to the two different therapeutic strategies that we use. First of all, the mutation-independent mini-gene augmentation therapy. So in this therapy we try to deliver a smaller version of the ADGV1 gene. And second of all, the exon skipping approach. In this approach, we try to remove the specific exons that carry the mutations. The third phase of the research will be very exciting, as we will start testing if our therapeutic strategies are able to restore the visual function of our mutant zebrafish model. So we have finished step 1 and 2 of our research, and the experiments for step 3 are currently ongoing. And then the question is, what next? We aim to finish phase 3 of the research halfway next year, and by then hopefully we have some proof of concept that our therapeutic strategies work in the zebrafish model. And then? Currently we don't have any funding to continue our Usher2C research afterwards, as my contract ends halfway next year. But we have many ideas. For example, it would be essential to also test our therapeutic strategies in the human situation. So we could start cell studies using human cells or start organoid studies. Alternatively, we could test novel therapeutic strategies or expand on, for example, the exon skipping therapy and define novel exons that we could skip in order to restore the protein production. But research is very expensive. One organoid study could already cost up to 20,000 euros, and one whole year of research is easily costing 100,000 euros. Would it be up to me? I would love to continue this research with the ultimate goal to deliver a therapy to halt ADGV1 associated retinitis pigmentosa. For now, I would like to thank all my colleagues at Radboud UMC and the Radboud University in Nijmegen, because I could not have done all this research on my own. And also, I'm very grateful that Stichting Usher Syndroom, Stichting Uitzicht and Cure Usher made the current research project possible by funding it. And lastly, I want to thank you for showing your interest in our Usher2C research. If after this presentation you still have questions or comments, don't hesitate to contact us by using the email address shown over here. Thank you.